C1S is a serine protease that plays a critical role in the classical pathway of the complement system, which is a key component of the innate immune response. The complement system helps clear pathogens and damaged cells from an organism, promotes inflammation, and attacks the pathogen's cell membrane. As part of the C1 complex, C1S is essential for the activation of the classical pathway, which is one of the three main pathways that activate the complement system. Structure of C1S. C1S is one of the subcomponents of the C1 complex, which is the first component of the classical complement pathway. The C1 complex itself is a large, multi-protein complex composed of C1q, a recognition protein that binds to antibodies, usually IgG or IgM, that have bound to antigens on the surface of a pathogen or cell. C1R and C1S, two serine protease proenzymes that are responsible for the activation of the complement cascade. There are 2 molecules of C1R and 2 molecules of C1S in each C1 complex. Activation. C1S is synthesized as an inactive zymogen, proenzyme, and is activated when C1R, another component of the C1 complex, cleaves it in response to C1q binding to an immune complex. Function of C1S. Structure of C1S serine protease domain. The key functional part of C1S is its serine protease domain, which cleaves specific peptide bonds in other complement proteins, leading to their activation. Activation of the classical pathway. The classical pathway is initiated when the C1 complex binds to antibodies that have attached to antigens on the surface of a pathogen. This binding induces a conformational change in C1q, which then activates C1R. Activated C1R cleaves and activates C1S, turning it into an active serine protease. Cleavage of complement components. C4 cleavage. C1S cleaves C4 into C4a, which is a small peptide that diffuses away, and C4b, which binds to the pathogen surface. C2 cleavage. C1S then cleaves C2 into C2a and C2b, where C2a associates with C4b on the pathogen surface to form the C4b2a complex, also known as the C3 convertase. The C3 convertase cleaves C3 into C3a and C3b, further amplifying the complement cascade and leading to opsonization of the pathogen, and eventual formation of the membrane attack complex, the MAC. Alpha Lifetech can provide sutimlimab biosimilar, anti-C1S mAb, and other biosimilar antibodies. www.alpha-lifetech.com. info@alphalifitech.com